Welcome to this video. In this video, I will explain prokaryotic transcription in a very simple way. I hope you find it helpful. The synthesis of RNA using the information present in one strand of the DNA is called transcription. According to the central dogma, genetic information is stored in DNA, which guides the synthesis of RNA, and mRNA helps in protein synthesis. A segment of DNA that is transcribed to produce one RNA molecule is called a transcription unit. It contains three critical regions, a promoter, and RNA coding sequence, and a terminator. Unlike DNA replication, in transcription only one strand is transcribed. This strand is called template, antisense, or minor strand. The RNA sequence is complementary and antiparallel to this DNA template strand. The other DNA strand which does not serve as a template for transcription is called the coding or non-template or sense or plus strand. It called the coding strand because it has the same sequence as the transcribed RNA, except that RNA contains uracil instead of thymine. The synthesis of RNA chain like DNA chains occurs in 5' to 3' direction with the addition of new ribonucleotides to the 3' hydroxyl group at the end of the chain. The enzyme that catalyzes RNA synthesis is called DNA-dependent RNA polymerase or simply RNA polymerase. Unlike DNA replication, primers are not required here, and bacterial RNA polymerase is composed of many subunits. Two large subunits, beta and beta prime, two copies of smaller subunit, alpha 1 and alpha 2, and one copy of fifth subunits, omega, which make up the core enzyme. Other functional subunits such as the sigma factor join and leaves the core enzyme at particular stage of transcription, and when the sigma subunit is attached to the core enzyme, the entire complex is called holoenzyme. By convention, the DNA nucleotides encoding the beginning of the RNA chain is called the transcription start site, and it is designated the +1 position. Sequence in this direction in which the transcription proceed are referred to as downstream from the start site and the number accordingly. Likewise, this sequence preceding the start site are referred to as upstream sequences and a number in the opposite direction. Note that there is no zero position. For simplicity, the whole process of transcription can be split into three stages: initiation, elongation, and termination. Initiation of RNA chain involves three steps. Promoter recognition. First, the holoenzyme searches for the promoter. The bacterial promoter contains an AT-rich region at the -10 position called the Tata box and a sequence at the -35 position. The initiating nucleotides, which is nearly always adenosine or guanosine. This structure is called closed promoter complex. The enzyme binds more tightly, unwinding bases nearly -10 region. When the bound polymerase causes local denaturation of the DNA duplex, it forms an open promoter complex. These steps require the sigma factor, and the bacterial RNA polymerase unwind approximately 17 base pair of the DNA, providing a template strand free to base pair with incoming ribonucleotides. The holoenzyme remains bound at the promoter region during the synthesis of the first 8 to 9 bonds. Afterward, the sigma factor is released and the core enzyme begins to elongation phase of RNA synthesis. The elongation phase begins after the formation of first phosphodiester bond. RNA polymerase stays bound to its template until the termination signal is reached. About 70 base pair of DNA are unwound throughout the elongation phase. The rate of elongation is about 50 nucleotides per second. The newly synthesized RNA forms a hybrid helix with the template DNA strand, about 8 to 9 base pair long. The region containing RNA polymerase, DNA, and newly growing RNA is called transcription bubble, containing locally melted bubble of DNA. The 3' hydroxyl group of the RNA in this hybrid helix is positioned to attack the alpha phosphorus atom of an incoming ribonucleotide triphosphate. There are two types of transcription terminators in E. coli. These terminators have two common features: a track of 7 to 10 consecutive adenosine and thymine rich region with A on the template strand and a GC rich segments with a palindromic sequence upstream of the AT rich region. This guanosine cytosine rich segment forms a hairpin structure, causing the RNA polymerase to pause and weaken its binding to the transcription bubble, leading to release. And in this case, it requires the Rho factor. Rho is a hexameric helicase that unwinds RNA-DNA or RNA-RNA double helix by translocating along a single strand of RNA in the 5' to 3' direction, and it is powered by nucleotide hydrolysis. RNAs with the Rho-dependent signals have a sequence rich in cytosine residues and poor in guanosine residue, including an upstream RAT site. Rho attaches to newly synthesized RNA at its RAT sequence and translocate along the RNA until it encounters RNA polymerase, unwinding the RNA-DNA hybrid helix and releasing the RNA. So that's all for now. I hope this video clarifies your understanding of bacterial transcription. If you are watching my videos for the first time, please press the subscribe button. Thank you for watching.